morning and thank you. Maddie's developing a punctal plug drug delivery technology for sustained drug delivery. The first step in this whole process is really designing a plug that stays where it needs to stay. And when we started this program, our first focus was traditional designs, and we used an arrowhead design to start off with, and quickly found that we could not get the retention rates that we were looking for. So we started working with anatomical models and then closely developed designs that more mimic the natural anatomy of the canalicular space. And that led us down several iterations of L-shaped designs, which are shown on the slide, until we finally got to the current design we're using, a straight L-shaped design at the bottom. We've tested this in over 150 patients in multi-center U.S. clinical trials. And in the last two studies, the GLA-12 study, which had a duration of 12 weeks, we showed a 96% retention rate over that period of time. And in the GLA-13 study, we showed a 92% retention rate. We also did extension studies that went on for an additional 8 weeks, and in that extension trial, we showed a 97% retention rate. So our overall retention rates with our current plug design are really favorable. The insertion process is very simple. It can be done at the slit lamp or just leaning the patient back. The insertion tool is put in the elbow of the plug, apply some pressure directly to the patient, and it's quickly inserted. You don't need a drop of proparacaine. It's a quick, simple procedure that can be done by ophthalmologists or even their attending staff. The initial insertion does require the patient to be dilated, but repeat insertions don't require that at all. The other thing that differentiates our device from other technologies that you've heard about today is there's only a one-directional drug flow out of the plug. We have a solid polymer matrix core, and then we put an adhesive on one end of that, and it's married with a traditional kind of silicone that's used in the punctal plug itself. The drug can only elute out of the surface, so it goes directly into the tear film where you want it to go, and it doesn't elute out the sides or go down in the canalicular space, so you reduce the overall level of systemic absorption, which potentially could lower our overall side effect profile. There are a number of different classes of drugs that can be formulated with this technology. In red shows the drugs that we're currently working on. Our lead program is latanoprost in glaucoma. We've studied that in a phase 2b study, which is ongoing. We've also studied olopatadine in phase 2, and we're currently working on nonsteroidals that we plan on taking into phase 2 next year, and then the step after that will be to work on steroids. So 4 different classes of drugs that we're currently working on or that we have entered in the clinic. This is a slide that I showed last year at this meeting. This is change from baseline and intraocular pressure with latanoprost and glaucoma, and we see a consistent 5 to 6 millimeter decrease in pressure going out over the 12-week follow-up period from the GLA-12 study. On the side effect front, this is a summary slide looking at 421 patients who have had the 95 microgram formulation, our current formulation, or less. The most frequently reported adverse event is increases in tearing. That level's at 13%. It tends to be mild in nature and usually resolves within the first week or so of treatment. I'll point out the rate of conjunctival hyperemia is about 7.5%, and contrast that with the 15% that's located in the Zalatan package insert. So overall, we think we have a high plug retention rate, a pretty good clinical efficacy, and a very favorable safety profile. Over the course of the past 6 months or so, we've been looking at the flexibility of this platform and started looking at additional formulations. The lower line is our latanoprost formulation in green. That's more of a really steady state type of elution. And we've been focusing more recently on travoprost in the red and blue. And we've looked at boosting the drug loads through higher elution rates early on in the elution mix for this. And then we asked ourselves, well, does that really translate into any kind of clinical benefit? We've tested that in a canine model, and this is looking at change from baseline and intraocular pressure. There's a control group in orange at the top of the graph. The purple line is basically Travatan Z eye drops, and then the blue line at the bottom is our extended formulation of travoprost. I take a couple points from this slide. First, the maximum IOP difference that we're seeing is in the 7 to 8 millimeter range, which is much more exciting than what we were initially seeing with travoprost or with latanoprost. The second piece is that the trend seems to be favoring the sustained delivery over the eye drop, throughout the course of this study. So we're very excited about taking this program into clinical studies next year. The other thing that we like about this technology is the flexibility that it can provide to treating physicians. You can envision a day where you place a travoprost insert in the lower tear duct of the eye and a complementary drug in the upper tear duct, a beta blocker, alpha agonist, or so forth. A post-surgical product could be a surgical placement of a steroid in the lower tear duct and maybe a non-steroidal in the upper tear duct. So there are a number of different combination approaches that physicians could ultimately use with this. We've been working on non-steroidals over the course of the last few weeks and months, and this is a graph of elution rate over time. And we're able to customize our elution profile depending on what we're trying to achieve. In glaucoma, we're interested more in a steady state elution profile, but for this product, which would be used in the surgical setting, we were interested in having a high initial drug burst, which would correspond to the immediate post-surgical time period. Our initial target elution rate for this product is 3 micrograms per day. We're able to achieve that with a high burst and then a quick diminishment to a steady state level that would carry the patient through the post-surgical period. Our IP position is one of Maddie's strengths. Since I was here at this meeting last year, we've added a new patent every 2.5 weeks on average. We've added 22 additional patents this year. There are 62 patents that have issued and 106 patents pending. We have a broad geographical coverage in our overall IP portfolio. Our pipeline, our lead program is latanoprost in glaucoma in a phase 2B study. We're planning on moving travoprost into a phase 2 study next year. We've already studied olopatadine in phase 2. We have a mixture of anti-inflammatories, both non-steroidals and steroids, that we plan on initiating clinical studies next year. I'll close with just summarizing some of the benefits that we see with this technology. First, we've already demonstrated there's a wide range of compounds that we can formulate. We're currently working on 4 different classes of drug. There's a flexible drug delivery profile, meaning if we want to have a burst, we can elicit that. If we were looking for more steady state elution, we can do that as well. It's a truly non-invasive approach. We're not touching the eye in any way. We're outside of the eye in the punctum. And we get, unlike eye drops, there's a relatively steady elution, meaning we don't have to have the high drug loads that eye drops need to overcome the tear turnover that naturally occurs in the eye. Our plug has been shown to be comfortable. We have excellent retention rates in the 92 to 96% rate in our last couple clinical studies. This is preservative-free. It's a passive system for patients, meaning the physician controls their compliance. And there's a reimbursement paradigm shift that we think will occur since there's a procedure fee associated with this. We have strong IP protection and a reasonable cost of goods. Thank you very much for your time.